Hi class, my name is Ruth and this is going to be my final project or presentation for our Ethics in a Digital Age class. It's very exciting. We're at the end of the semester and I'm looking forward to reading or watching everybody else's final project. But my project is based on the promise versus the controversy of the CRISPR Cas9 technology that's emerging. So there's about 15 slides in my presentation, and basically you'll just click through every slide and there's a little microphone button, and when you press it, I'll basically talk through each slide. I hope you enjoy. So as you all probably know, our bodies and our cells are all made up of DNA. DNA itself is millions and millions of pairs of DNA which create a language or a blueprint of who we are as a person or as an individual. And within that language or blueprint, there are certain regions which code for our genes. And those genes are incredibly important because they make up everything about us and they are responsible for our general health and how we live every day. Examples of our genes would be our hair color, eye color, height, weight, even genetic dispositions for healthcare or health issues, such as heart disease, cancer risk, high cholesterol, and things like that. So what is CRISPR? So CRISPR is an emerging technology that can generally be used to edit genes. It's a gene editing tool. It stands for clustered, regularly interspaced, short palindromic repeats, which is a mouthful to say and certainly not short at all, or even easy to remember. So how does CRISPR actually work? This next slide will show the science behind this technology and how it targets a specific area in our DNA to edit, but I'll review it step by step. So the first step in gene editing is that the scientists identify a gene responsible for a specific trait. They then create a piece of the RNA, which is part of our DNA, and they create also an enzyme to target and edit that specific gene. The RNA that is created then mimics the end of the targeted gene's DNA in an enzyme, which is called Cas9, which cuts the DNA and introduces that to the cell. The RNA then locates and binds to its DNA sequence, and the Cas9 enzyme is what's responsible for cutting across both strands of DNA. This results in a change in the DNA or the mutation, and then a trait can be added by removing the gene or introducing a new one. After this change occurs, the scientists would actually rely on the cell's own ability to repair its DNA sequence, which is automatic. It's a little confusing to figure out the exact science behind this because there's so many steps and there's so many terminology that's used. But just imagine this picture in the next slide and it'll give you a general picture of what CRISPR is supposed to do. There's also another slide after the next slide, which is just a general picture that you should keep in your mind of what CRISPR does. And it's the picture with the hand that's holding a scissor and it's cutting the DNA. And that's basically what CRISPR is all about. It, in theory, it will cut a piece of the DNA, cut that part out, and allow scientists to put a new part of the DNA in, or a part of a gene into that DNA to edit it. So what is promised with the technology of CRISPR? This next slide just generally lists all of those promises. But scientists claim that parents would eventually be able to choose traits for their children, physical traits, or remove traits that are passed down from generation to generation. Also, with the increasing human population and the climate change in the world today, there is an urgent need for mass production of crops. CRISPR can help our agricultural system by creating plants that not only produce in mass amounts but can produce crops that are more nutritious and also resistant to drought and pests. In the next slide, you'll see an explanation of how CRISPR is supposed to help with production of drought-resistant corn, and that's just a visual aid. But CRISPR paves the way to potentially cure inherited diseases such as disease, cancer, and genetic disorders such as cystic fibrosis, sickle cell disease, and even Huntington disease, which are all caused by a single gene mutation. CRISPR also promises to allow researchers to develop gene therapies to fight infections caused by the human immunodeficiency virus, or HIV. However, with all of these promises and everything looks crystal clear and the grass is situation, there are definitely ethical and safety concerns that are certainly raised with this new technology. So the main ethical concerns that are listed here would be safety, uncertainty, informed consent, and the need for laws and regulations. CRISPR is a new technology, and because it is new, there is inadequate research, and there needs to be more published data in order to establish this new technology. I believe personally CRISPR will always be a safety concern because of the uncertainty of this technology. Researchers are basically given the freedom to manipulate a human's genetic code, which is a really big deal. And once that genetic code is altered, it's not just for that person, it's basically being manipulated, and these genes get passed down from generation to generation. So there is also a risk of disability, risk of other parts of the gene that are unintentionally edited, and all of these risks would be very, very difficult to control. This would also raise the concerns that if revolutionizes in vitro fertilization, they would allow their parents to choose their offspring traits, which creates the concern of who would be able to choose their traits and how guidelines would be created or how laws would be put into place so that there is control, without the fear of creating these future designer babies, as they call them. And the next slide shows basically the options would potentially get in terms of choosing what kind or what type or what specific traits their baby would possess. So this cute, perfect baby is something that CRISPR would promise to make, basically, in the future, if it ever comes to that point. They would be able to remove traits such as baldness, male pattern baldness that runs in the family, perfect pitch, like these type of qualities, 20/20 vision, perfect vision, and also types of diseases that are passed down or traits that are passed down from generation to generation. So this is what CRISPR promises to do. I certainly do not think our world is ready for this type of innovation, just because there's so many risks of safety concerns. There's so many risks of who would be able to choose this. What right do parents actually have to choose their babies? And not only just their babies, these traits would be passed down from generation to generation. So if there is a mess up or there is a mistake that happens in this gene editing, it not only affects that baby, it affects the future babies of that baby. And it's interesting to see how this would all turn out if it ever comes to this point. All right, so these next two slides basically show how genes are edited with the CRISPR technology. In August of 2015, a group of Chinese researchers, led by an oncologist at Sichuan University, started testing cells modified with CRISPR on patients with lung cancer. And what they did was extract immune cells from the patient's blood, and they used CRISPR to add a gene sequence which will help the patient's immune system target and destroy the cancer cells when it is reinfused or reintroduced into the patient's bloodstream. So the next slide after this is another simplified version of this slide, which better explains how this tool is editing these genes in cancer patients. So in recent news, in 2018 actually, this man, this Chinese researcher, He Jiankui, he claims to have implanted CRISPR-Cas9 embryos into 6 women, which resulted in 1 successful twin pregnancy and 1 single pregnancy. He claims to have disabled a gene that is responsible for protecting the body from an HIV infection. However, when they found out that he was actually doing this type of study, other researchers claim that he may have caused mutations in other parts of the twins' genome, which could have severe health consequences for these children. Unfortunately, only time will tell. It's only been a year and a half since this all came out. But he has definitely been criticized, but not just because he pursued this illegally, but he neglected to do adequate safety testing, and he also failed to follow, standard procedures of a clinical trial. In short, he was investigated. He was fired from his position as a professor at his university, and potentially could face criminal charges depending on the outcome of these twins. This example just shows how this technology needs to be regulated because of the controversial issues that arise from it. The next slide shows the risks versus the benefits of CRISPR, which will go more in detail. All right, so here I've just listed the risks versus the benefits, and I've made it equally in terms of listing the risk and benefits, just so I can keep a standard ground. So you can read here that the risks are that it's controversial. Generally speaking, this new technology is so different in terms that it can alter our human traits and our personalities and the way we look, which will eventually be illegal in some countries, I can assume. There are privacy and confidentiality breaches, which I'll go more in depth. So this brings back to the previous slide that one of the main ethical issues would be obtaining informed consent. It's the ethical issue that obtaining a consent from the future offspring that they're trying to edit is impossible because the parents already made the decision that affected their future children. I have worked in infertility before, and we have seen so many emerging technology such as PGS, which is called pre-implantation genetic screening, and PGD, which is pre-implantation genetic diagnosis, which basically is all technology that helps increase the chance for a woman to have a healthy implantation when she is undergoing in vitro fertilization. So this technology screens for extra or missing copies of chromosomes, while also looking for gene defects and genetic disorders. I believe researching the process of CRISPR would raise this ethical issue for a world that may or may not be ready to accept gene editing. It's hard to obtain informed consent from this future offspring because it's almost like the parents are just allowing this to happen without taking into effect of how it could affect the future generations. Um, there is a risk of serious injury, disability, and death, um, and also the risk of misuse. If it ever got to the point where this technology got into the wrong hands, who's not to say that they will create, super babies with high IQs, like in the previous slide with the baby, and just all these traits that can be changed. Um, and as well as the fact that it's difficult to regulate and supervise. The National Institute of Health, they have called for an ethical discussion to take into place on CRISPR, because they need more information and it needs to be established and there needs to be more studies published. So it is kind of comforting to know that the scientific community is already concerned with this ethical issue and also the safety concerns. Um, but the benefits as well, going over that, it is definitely a new technology. It definitely advances where we are right now in terms of genetic tools or genetic editing tools. It promises, obviously, like we talked about, the disease and infection control. If at one point they are able to, for example, help mosquitoes or help create mosquitoes that ward off malaria or they don't carry malaria, that could be a great innovation for certain countries who are infested with that. Immunotherapy for cancer patients. There are so many cancer patients suffering right now, and working in oncology right now. I do take it on a professional level and a personal level. All these patients that are suffering with chemotherapy and radiation therapy, the side effects of that alone, if it could be, changed with altering the DNA of cancer patients and altering the genes in order for our bodies or our own immune system to attack our cancer cells, I'm sure that the side effects would be much less, rather than having, poison such as chemotherapy being injected in your body or being, sunburned with the radiation. I'm sure that in terms of immunotherapy, it's our own immune system attacking the cancer cells, so I'm sure that the side effects would be much less. Crop cultivation, like we talked about the crops that are drought resistant and pest resistant, those are all benefits for mankind. So it's not necessarily the risk outweigh the benefits or the benefits out. They're all listed there. They are present and they are there, and it's up to researchers and our lawmakers and our government to be able to come up with a policy or a regulation or laws, in order to make sure that this technology is safely researched and is safely administered or safely done. So with all the ethical frameworks that we have discussed and reviewed this semester, I believe that the consequentialist ethical framework would help establish a guideline or a policy for this new emerging technology. So as everyone remembers, consequentialism allows a person to look at the consequences of something before acting on that. Like the promises of CRISPR, consequentialism makes it difficult to know the result of that action or what the result of what CRISPR would be. This would allow our government to make laws and regulations based on whether it would affect the greater good or our society as a whole, and whether it would affect it positively or negatively. There's always risk to every new technology that is out there, not just CRISPR, and that risk of this technology, is whether it's going to be abused or misused is something that lawmakers and researchers have to take into consideration. There needs to be more research studies and clinical data published, not only in just animals, but also in humans. Human clinical trials need to be established. And then regional regulations and laws have to be created, and they don't need to just be lawmakers or government officials. They need to include the researchers who are actively participating in these trials and who are actively researching this on an everyday basis. It needs to come together. And in terms of proposing a policy, they need to outline guidelines for safety. They need to guideline specifically um, what trials or what limitations there are for each trial. It's not a free for all. Well, we'll test all these cancer patients and this who have this diagnosis. It needs to be more in control, and it needs to be established just like every clinical trial that's out there right now, specifically for cancer patients. I believe that CRISPR would deliver on much of its promises, and it probably will happen more quickly than we all think. But I believe that it'll take longer for the public to accept this new technology. It is so new and it is so different that it would take a while, and especially with social media nowadays, it would definitely take a while for our society to accept this technology. Not a lot of people know what in vitro fertilization is, and that technology is only less than 50 years in the making, and even up until now, there's people who don't know what IVF is and in vitro fertilization is. So I believe that it would take some time for CRISPR to make its way into our world, especially with the world that we live in today. There's a lot of personalities and a lot of opinions online right now on our social media. So I definitely think that it will definitely be a challenge for this new emerging technology to arise. But like every great technology, it takes time, and especially something that can harm us as individuals or harm our society. I think it's important that scientists take it slow, just because there's so much important factors to take into place. And CRISPR is definitely a technology that I would love to see moving forward, but there would definitely need to be proposals and policies in place, and they all need to rely on the ethical frameworks that were first established, and they just need to worry about the safety of people in general. All right, guys, so that is my final project. I hope you enjoyed it. I know it's a little weird with the PowerPoint and everything, but I hope you were able to get through it very quickly and you were able to understand it. And I hope to look forward, and I'm looking forward to watching and listening to all of your final projects. All right, thank you guys. Have a great day. Bye bye.